Welcome. This is the second of three presentations designed to help you prepare for the biology MTELs. In this presentation, we'll look at some more of the sub-areas of biology, and then we'll go through some sample questions. If you click on the hyperlink details on the downloaded presentation, it'll take you to the DESI MTEL site, which will go into more details of the kind of specific topics within these different sub-areas. But just as an overview, with the last presentation, we covered the chemistry of biology as well as molecules. With this presentation, we'll be looking at cell metabolism, cell structure. We'll be looking at human anatomy and physiology. We'll look at some areas of ecosystems, how they're structured, how energy flows through ecosystems, how humans affect ecosystems and the environment. And we'll look at also genetics, heredity, and evolution. Okay, so let's start our journey through some of this subject matter. As we're moving through, you might find that some of these topics feel a little disjointed or kind of popped in as afterthoughts. I've looked for opportunities to put materials, definitions, concepts into the presentation if I've encountered them in materials, in MTEL questions, just so that they're kind of covered and highlight that they might be something that you'll encounter. And as we're going through, it's less about a comprehensive review of biology, right? That would be impossible in 3 presentations. But a kind of navigating journey through these different sub-areas to get a sense of where you're at with them. Okay. In terms of cells, right, cellular energy will definitely be something that you'll encounter, right? So you can think about this at the cellular level, how are cells making energy, right? Or kind of the broader life system level, right? So life is inefficient, metabolic processes generate heat, and there's loss of energy. So we need to constantly be moving energy into the system. And one way we can do that is with autotrophs that can take the energy from photons, right, and use that to drive something energetically unfavorable. That's the fixation of carbon from the atmosphere, right? Carbon dioxide, right, and being able to build organic molecules, right? That creating that order is not favorable, right? So, but photosynthetic organisms can do that. Right, so you can take carbon dioxide potentially from the atmosphere and water. You can use that with some sort of energy input like solar energy to synthesize organic molecules. So shown here is glucose, right? C6H12O6, right? And within this balanced equation, right, there's also the production of oxygen. So now to move in the other direction, right, organic molecules can be broken down, right? Energy within the covalent bonds that build them, right, can be exploited and used to convert into different types of molecules. Right, so here with the oxidation or the breakdown of glucose, right, within cells, right, they can use that breakdown of glucose or other organic molecules to be able to convert that into a chemical energy that's useful for the cell. Right? And the typical type that we talk about, right, is adenosine triphosphate. And with that process, we're also creating heat and carbon dioxide and water. Right? So this is a nice kind of cycle. Okay, carbon dioxide and water. Within photosynthesis, we're splitting the water and producing oxygen, right, but also building organic molecules like glucose, right, and then those can be broken down to produce ATP, and then there's also these other products, right, water and carbon dioxide. So let's take a moment and think about plants, right, a photosynthetic organism. So just want to highlight some of the differences between plant cells and animal cells, right, both eukaryotic cells. But plant cells have vacuoles, these kind of storage organelles that can also be used for osmoregulation. And then also a reminder that plants have cell walls, right? So which have some integrity and structure, right, and contain a significant amount of cellulose. In terms of kind of plant cellular anatomy or tissue anatomy, a reminder of these structures called stomata. So they have these guard cells, and they can open or close this structure. And when open, it allows for the movement of carbon dioxide, right, from the atmosphere into the plant tissues. But also water can escape through this structure. Photosynthesis we can break down into two different kind of stages. So there's the light-dependent reactions, which depend on light, right? And then there's also the light-independent reactions. Light-dependent reactions, what's shown here is a chloroplast, right? One of these double membrane organelles. Within the chloroplast are the structures and the molecules that can take photons, right, and funnel them into pathways that can be used to create ATP and NADPH. Right, so this is basically energy, right, and then also high energy electron carriers, NADPH. So these can then be moved into the reactions in the cycle, the Calvin cycle, and it's a Calvin cycle that actually takes carbon dioxide, right, can integrate them into organic molecules within the Calvin cycle, which can produce G3P, which can be then converted into glucose. Okay, so this is part of the carbon fixation cycle, the Calvin cycle. So now that we have the building of glucose, right, so now we can think about taking organic molecules and breaking them down to make energy, maybe within this organism, right, or an organism that consumed it. So that brings up cellular respiration. So cellular respiration can be thought of as potentially three different stages. So it's how a cell is taking an organic molecule and making energy, making ATP, right? Because ATP is adenosine triphosphate. It's that molecule that many enzymes can use to be able to fuel their generally unfavorable kind of processes, right? So it's what we call the energy currency of cells, ATP. So how is a cell making ATP? How are we taking an organic molecule and converting that into ATP? Right? It's cellular respiration, and the molecule that we tend to talk about is glucose. So of the stages that could potentially happen with cellular respiration, you have glycolysis. That's the breakdown of glucose, right? It's right there in the name. Glycolysis, right? Glycolysis. To lysis is to loosen or to break down. And glycolysis, the breakdown of glucose, actually produces a little ATP. So we get the net production of two ATP molecules, right, when we start to oxidize glucose, right, and convert that into two molecules of pyruvic acid. And it's a good thing that we can create a little bit of energy there, right, the 2 ATP, because this process doesn't require oxygen. So even if there isn't access to oxygen, a cell can still do the first step. It can still do glycolysis, and it can still make 2 molecules of ATP. Not very much, but it's something. Now, if oxygen is present, right, the product from glycolysis, pyruvic acid, can make its way into the mitochondria, right? So glycolysis, this is something that comes up, right? Glycolysis happens in the cytosol, right? The rest of these different stages happen within mitochondria. The pyruvic acid, right, gets transported into the matrix of the mitochondria and then gets funneled into the citric acid cycle. And the citric acid cycle also makes a little ATP, but importantly, it also produces NADH and FADH2. These are what we call high energy electron carriers, and they're making these high energy electrons, or at least carrying them, to be able to funnel those into the electron transport chain, right? This third kind of component of cellular respiration. As the electrons make their way into the electron transport chain, these hydrogen pumps move hydrogen ions into the space between the membranes of the mitochondria. They concentrate the hydrogen ions there. So the hydrogen ions, of course, are dying to get out, right? Things want to move from high concentration to low. And fortunately, the ATP synthase allows them to leave that space. The hydrogen ions can flow down away from where they're highly concentrated back into the matrix of the mitochondrion. And as the ATP synthase allows them to move through its structure to go from a high concentration to low, right, it uses that movement of hydrogen ions to synthesize ATP, right? And actually it produces an enormous quantity of ATP. So for each molecule of glycolysis, or for each molecule of glucose, within cellular respiration, right, we're producing 2 ATP early on, and that doesn't even require oxygen. And if oxygen is present, we move into the next 2 steps of cellular respiration that produce 2 molecules of ATP with the citric acid cycle, right? Potentially 32 molecules of ATP with the electron transport chain, right? And again, that's if oxygen is present, right? So that's where an enormous amount of ATP can be produced. Oxygen is the final electron receptor in this whole process, right? So this is where the oxygen piece is required, right? It's at the very end of the process, right? And we get the synthesis of water. So if oxygen is not present, right, that leaves us with something of a problem, right? Because now we're going to get the buildup of pyruvic acid. So in the context of not having any oxygen present, right, looking at this pathway on the left, right, if we start to get a buildup of pyruvic acid, right, we've got to do something with it. So with fermentation, right, that occurs differently depending on context and organism, right, but pyruvic acid, right, or pyruvate can be converted to lactic acid, right, and then we can get a renewal of NADH, right, back to NAD, right, or pyruvic acid can be converted to ethanol and carbon dioxide, right? So there are these 2 different approaches where we can take the buildup of pyruvic acid, convert them into a different type of molecule, and also renew our source of NAD. So with cells in the cell cycle, right, as well as mitosis, we can break the cell cycle down into these various different stages. Shown in left is mitosis, so that's the series of stages we're all familiar with where you get a cell dividing, right? The period in between mitosis, where a cell isn't actively dividing, we call interphase. Interphase can be broken down into a series of phases, right? G1, S, and G2. And they naturally make sense in terms of what these different phases are responsible for. If you think about them in terms of what a cell is going to need to deal with if it's coming out of mitosis or getting ready to go back into it. Importantly, I should highlight that there's also G0, right? There's this resting phase, which is where most cells in our body exist, right? They're not actively dividing. They've assumed some sort of a function. If you think about a neuron in a brain, for example. But if cells are actively dividing, they're going through the cell cycle. G1 is going to be an opportunity, right? This is cells coming out of mitosis to make sure that mitosis progressed correctly, the chromosomes have segregated the way that they should. Right? It also gives these cells, which are now probably about half the size because they just split from 1 into 2, right? Need an opportunity to rebuild some of these different molecules, to grow, to duplicate things that were lost through cell division. Right? So this is all occurring during G1. There's also checkpoints in these different types of phases. And if something hasn't occurred correctly, it gives a cell the opportunity to correct it or to undergo programmed cell death, apoptosis, and take itself kind of out of the equation. If there's a problem and the cell could be problematic for the benefit of the organism, they can undergo programmed cell death. S phase, when I think S for S phase, I think S for synthesis. This is where a cell is duplicating its DNA because of course it's going to need to duplicate it, right, if there's a round of cell division coming up. So S phase is for synthesis, right? It's where you get a duplication of the DNA. G2, right, is going to be the second kind of gap phase. And following S phase, it makes sense that the cell needs an opportunity to make sure that the replication of the DNA and the fidelity of that replication was high, right? That there aren't errors within replication. If there are, the cell needs opportunities potentially to correct them. If not, potentially to undergo apoptosis, right? And the cell is also gearing up in preparation for another round of cell division. Within cell division mitosis, we have the PMAT stages, right? There's prophase, metaphase, anaphase, telophase, right? And then cytokinesis. Prophase and metaphase sometimes have a stage later in prophase, depending on the reference, right? Called prometaphase, right? Or late prophase. So looking at these different stages of mitosis, right, we have interphase where, DNA replication occurred. So now there's duplicate sets of chromosomes. The cell gearing up for mitosis first enters into prophase. And these stages will make sense if you think about also, like, sometimes it's nice to kind of take a step back and just think rationally, all right, how is this going to occur, right? It made kind of sense with the different phases of the cell cycle. How is this going to occur with mitosis? With mitosis, right, the first thing we need to do is we need to take these chromosomes, right, which had been relatively, they're decondensed. They look almost like a bowl of spaghetti or something like that. If we are going to try to segregate these chromosomes into daughter cells, right, it would be a real challenge. So the chromosomes condense, right? They turn into these discrete little packets, right? So that occurs early on in this process, right? And the artist has shown that with the relatively decondensed chromosomes, right, moving into these condensed chromosomes, which kind of take on that morphology like we're used to looking at them in a textbook, where they look almost like an X, right, because they're duplicated. We need to move the centrosomes, right, these organizing centers, to opposite ends of the cell. So that also occurs early in mitosis. How on earth are we going to move chromosomes around within this cell that's actively dividing and ultimately into daughter cells on opposite ends of the cell if they're contained within a nuclear envelope? Well, you can't. Right? So it also makes sense that late in prophase, right, or in prometaphase, you get a breakdown of the nuclear envelope. And this is one of the things actually that amazes me with cell bio. There's all these little kind of random processes that don't seem like they're all that interesting, but turn out to be like incredibly fascinating. Where you get the nuclear envelope, right? This membranous structure that surrounds the chromosomes. It begins to kind of disassemble, but you've got to reassemble this monster in not too long. So it's not a totally chaotic just destruction of the nuclear envelope. It ends up being this kind of transient, intermittent mitotic stage where it can be reassembled, right, within a few hours. But you get this kind of deassembly of the nuclear envelope, right? And then we move into metaphase. So at this point, the mitotic spindles have formed, right? So we have these microtubules extending out from the centrosomes, and they're interacting with the chromosomes, right, at the centromeres. And with these mitotic spindles, it's like a tug of war where the microtubules are dynamic and they're pulling on the chromosomes. They're pulling on these structures. And they're both pulling about with equal tension. So as a result, the chromosomes line up along the middle. It's M for middle, M for metaphase, right? This is the metaphase plate. This is where the chromosomes are all nicely aligned along the middle. But these sister chromatids, right, the duplicated chromosomes, they're adhered to each other. So we get this pulling and tension to each pole of the cell, but because they're adhered, they kind of line up along the middle. Right? This structure releases, right? So the sister chromatids can now be separated. And when I think anaphase, I think A for anaphase, I think A for away, right? The sister chromatids move away from each other towards the opposite poles. Right? And then we're in telophase, right, or telophase. So this is now where we have the sister chromatids, right? They're now on opposite sides of the structure within the presumptive daughter cells, right? We can now get the reassembly of the nuclear envelope around these structures, right? And then the decondensation of the chromosomes, right, as we move back into interphase with these daughter cells. All right, a reminder of the central dogma, right? So the central dogma is one of these core unifying concepts of life, right? So the initial code of life, right, as we call it, is within double-stranded DNA, right, within these nucleic acids. Within the DNA, there are discrete coding regions. Most of those code for proteins. Within humans, there's roughly 20,000 genes within the double-stranded DNA, 22,000. If those genes are destined to be expressed as protein within a cell, right, this is where transcription occurs, right? So sometimes I think about DNA as being like a library, right? And within the library, you don't check out all of the books all the time, right? Within the library, you just kind of check out what you need in any given context. And that's kind of what a cell does. It kind of checks out whatever genes it needs, or turns them on, or expresses them. When it does, right, we call this transcription, where the double-stranded DNA gets transcribed into messenger RNA. So messenger RNA, also nucleic acids, is single-stranded. And it's a reflection of the codon, right, the triple nucleic acid codon language within the double-stranded DNA, right? That's reflected within the messenger RNA. And then that, 3-letter codon, so these codons, right, they encode for one of the 20 amino acids, right, or a stop codon to stop translation, right? They're present within the messenger RNA. Oof, rough. Okay, a quick review of the central dogma. Central dogma is a way to think about one of these unifying concepts of life, where you have the code of life is within DNA, right? Double-stranded DNA contains genes. If those genes are relevant and activated and destined ultimately to see the light of day as a protein, right, these genes get transcribed into messenger RNA, also nucleic acids. And then the messenger RNA, right, gets translated into proteins. And then proteins, in most cases, right, are the little molecular machines that are doing the work and providing structure and communication within the cell. A comment on anatomy and physiology. So human anatomy and physiology is certainly something that you'll encounter. So if this is in a course that you've taken, or maybe you haven't taken it since high school, right, this could certainly be an area to focus on with some of the references that I've suggested, or the book you can borrow from me. But human anatomy and physiology talks about the macronutrients, and in some cases those are the 4 classes of organic molecules that we talked about with the first presentation, as well as the digestive system. Right? And then it breaks the body systems down into circulatory and immune, right? Respiratory and excretory. Excretory refers to the urinary system. I don't know why they call it the excretory system. That's very atypical. The nervous, reproductive, and endocrine systems, right? The muscular and skeletal systems together, and then the broad kind of concept of homeostasis. And so homeostasis is this idea that generally, right, cells, tissues, organisms, the organism is in a favorable state. This is the assumption, is in a favorable state, and a deviation from this state needs to be corrected. So any deviation needs to be opposed, right? So usually what you see is negative feedback, right? So don't think negative is down. Negative means opposing. So something goes up, right? Got to bring it back. Something goes down, got to bring it back up, right? So this is this idea of maintaining a similar kind of state, homeostasis, through negative feedback. The classic example there is with glucose levels in the blood, right? So as glucose goes up, let's say it's after a meal or something along those lines, right? Insulin is a hormone that's secreted, right, by beta cells in the pancreas. Insulin helps to promote the movement of glucose into body cells. And as it does that, it moves glucose from circulation, from the blood, right, into cells and tissues. As glucose goes down, right, normally this needs to be maintained, right? We need to maintain glucose levels after meals, but also in between meals, while you're resting, right? These need to be maintained. So when glucose levels go down, the hormone that takes action is glucagon. So glucagon, secreted by alpha cells in the pancreas, in islets of the pancreas, helps to bring glucose levels back up. It does things like break down the storage form of glucose, glycogen, in muscles and the liver, break it down and release it into the blood to bring glucose levels back up. So this is a classical case of negative feedback. Right? Another favorite textbook example is body temperature. Right? When you're cold, you're going to shiver, right? And you'll get vasoconstriction of vessels on the surface of the body. When you're warm, right, you get vasodilation, right? You start sweating. These kind of opposing responses to maintain body temperature. All right, moving on to ecosystems. We can think about ecosystems as a way that they're structured. So with ecosystems, you'll certainly encounter this idea of trophic levels and energy transfer. So at the base of what we can think about is like a pyramid with a structure of ecosystems. Right? You could have decomposers, right? So these are things that are breaking down detritus. So they're going to make up a significant amount of the biomass within this particular system. This becomes kind of more refined, reflective, and predictable when we get into looking at things like primary producers, right? Primary producers would be like the autotrophs that we looked at with photosynthesis. So they'll make up the most substantial amount of biomass within this kind of formulated pyramid representation of the structure of a community within an ecosystem. Primary producers, right, can be consumed by consumers, primary consumers, right? So now we've talked about heterotrophs before, now we're bringing them into this idea of a community within an ecosystem, right? And then these can be consumed by secondary consumers and so forth. With each move between trophic levels, right, there's only about 10% of the energy makes its way through trophic levels, right? And this can be reflected in part by the biomass. So there's going to be a loss of energy between these different levels as heat, right, as waste from metabolism. So that's a concept that you will encounter probably in multiple choice or maybe even in open responses. In terms of the nature of populations, right, we can look at a distribution of ages within a population. And from that distribution of the ages of a population, right, and here, right, this could be reflective of human population, right, but it's going to depend on the organism in terms of age, because you're also thinking about when reproduction, right, is brought into this concept. if you look at a distribution of males and females within a population, if there's a disproportionately high number of members of that population that are of the reproductive age, right, there's going to be an increase in population, right, within the subsequent generations. Right, so that can be shown with rapid growth, right, or slow growth, right? Something stable would be if there's kind of a transition of a population into an kind of a more balanced abundance of members of the population of reproductive age, that could end up kind of stabilizing the population. Or if there's a decline, right, in reproductive members, right, then you're going to see a decline in that population number. There's various different ways that organisms can interact with each other, and we can define some of these in terms of kind of a symbiosis of these different organisms, and we can define these in terms of who's benefiting, who's not, or maybe there is no effect. Mutualism is a case where both partners within the symbiosis benefit, right? So here it could be flowers and bees, right? The flowers get pollinated and the bees get nectar, right? Everybody's happy. Commensalism is a case where one of the partners, right, in the partnership, right, is benefiting, right, but there's no effect on the other, right? It's not a plus or a minus for the other member of the partner. So in this sense, it kind of, particularly for that one partner, just kind of commences. It is. And lastly, parasitism is when one partner within the relationship benefits, and it's to the detriment of the other. Or you could also think about this as a case of kind of predation. So in this relationship, one benefits and it's detrimental to the other. Thinking of tiers of how you can think about from the simplest to the most complex. We can look at the structure of ecosystems really and think about them in terms of who's playing a role or who's playing a part or who's the focus of a conversation or a study, or just a way to think about or talk about different dynamics within an ecosystem. So if you're just talking about an individual, right, that's an individual organism. If you're looking at those individuals within an ecosystem, right, of the same species, right, this is the population. We can look at the structure of ecosystems by thinking about kind of, there's different levels of complexity to think about ecosystems. Maybe that's meaningful for a conversation or a research study or just a way to think about what's happening in terms of relationships within an ecosystem. An individual would be one organism, right? And many of those species, many of those individuals within a species would make up a population. An interaction between different organisms, different populations, right? This is going to be kind of the concept of the community. If you're also integrating in abiotic factors, like shown in the illustration here, you have a rock, right? This is going to be the entire ecosystem. Multiple ecosystems, not necessarily interacting, right, are going to make up a biome. And it's the biomes together, right, that make up the unified singular biosphere. We looked at the idea of structures of an ecosystem and the flow of energy between these different trophic levels. We can also assign some different kind of terms, where we have the autotrophs, right, were shown before as the photosynthetic organisms or the primary producers, which are situated on top of the decomposers, right, the detritivores. So above the primary producers, the autotrophs, the photosynthetic organisms, right, we have herbivores as primary consumers, right, and then we have secondary consumers, which could be carnivores, right, or would be carnivores, right, and then we also have the top carnivores, the tertiary consumers. This is another perspective of kind of a pyramid representing an ecosystem, right? So on the base of the pyramid, you have the detritivores, right, the decomposers, and then you have energy into the system through the photosynthetic organisms, the autotrophs. Those that are consuming the primary producers, right, are the herbivores, the primary consumers, right? And then right, you have the omnivores that could be consuming herbivores or other animals, right? And above those, you have the top carnivores. This is another representation of community structure, right, where you have the decomposers on the base. You have the photosynthetic organisms at the base of the pyramid, right? Consuming the photosynthetic organisms are the herbivores, right? These are the primary consumers, right? Above this, you have a kind of a mixture of carnivores and omnivores. You have those that are consuming the primary consumers. And then at the top, where you have the fewest, right, representative species, you have the lowest amount of biomass, right, is where you have the top carnivores. All right, lastly, genetics, heredity, and evolution. So if you're comfortable with Punnett squares, that's great. If not, you'll definitely want to practice drawing out some on your own. You could start with a simple, straightforward example, like the one on the left, right, where you have about as simple as it gets, right? So you have a homozygous recessive parent for some sort of a phenotype, right? It doesn't matter what the phenotype is. For this case, it's the color of a bean pod. And then on the left, you have a parent that's heterozygous. And then it's mapping out, pretty straightforward art. Each of these parents are going to produce a gamete, right? And that gamete is going to carry one copy of the allele. So it's going to carry one or the other. And depending on what it does carry, right, you can map out the outcomes. So in this case here, right, it would be 50% heterozygous, 50% homozygous recessive, right? So pretty straightforward. But again, right, if it's something you haven't done in a while, or you kind of forget how it works out, I'm always a big advocate for actually drawing it out by hand, trying a couple different kind of paradigms and just making sure that it flows pretty naturally. One case that I've encountered a bunch of different times, right, in the prep materials and questions is something that's a gene that can be sex-linked, right? So for example, just as a reminder, right, humans have 23 pairs of chromosomes. 22 pairs of those are the autosomes. But then there's also a pair of sex chromosomes, right? So that's XX or XY. Now, this is a case where it's interesting because it's not always the case where an individual has two copies of every gene, right? So in this case here, the individuals that are XX, right, are going to have two copies of every gene on the X chromosome. But individuals that are XY, right, will only have one copy. And that potentially brings up something problematic, right? So if there's some sort of a mutation or a recessive allele that is on the X chromosome, right? If you're an individual with two X chromosomes, that's okay because you have another opportunity to have a normal copy or wild copy of that particular type of gene. But if you're an XY individual, right, you only have that one X opportunity, right? If that X chromosome is carrying a mutation or some sort of a a recessive allele that potentially isn't beneficial, then you got it, right? Because that's the one opportunity that you have to have the gene is on that X chromosome, and it's the only one that exists. So it's worth taking your time to thinking about what the fallout of this is. And with these different arrangements here, so one example that's pretty typical is color vision, right? So I think 10% of males have some sort of disrupted color vision due to genes that exist on the X chromosome. And follow through with these different arrangements. This is all of the different arrangements that could be encountered, whether along the top here, if you're looking at normal color vision, right, paternal contribution, or right, a paternal contribution from an XY individual that's carrying the mutation. And then you can think about all the different circumstances where the other paternal contribution, the XX, could have normal, right? Could be a carrier, right, but has normal color vision, or is carrying this recessive variant, right, or this mutant version of the gene on both X chromosomes, right? So take some time to kind of work through these different scenarios to see what the outcomes are. Lastly, there's the dihybrid cross, right? So this is thinking about not just one gene, right, and the different alleles for that gene, but thinking about two genes and just going through the same process, right? So you have up at the top here, you have the parental contribution for two genes, right? And the initial cross would be homozygous dominant and homozygous recessive. So all of the F1 generation, right, would be heterozygous for both of those genes. Now, if you cross individuals from the F1 generation, right, you can do this dihybrid cross, right? So you can think about what are the different options for the outcomes for these offspring, right? And if you look at the potential outcomes, right, there's a ratio of 9 to 3 to 3 to 1, thinking of these two different genes and their outcomes kind of independently. And if you get this 9 to 3 to 3 to 1, right, it demonstrates that these genes are working independently, right? They're predictably behaving as they should with independent assortment. There's no kind of interaction between the two genes. That would be what you would expect to see. Now, if there's a deviation from that phenotype, it would suggest that there's some sort of an interaction or association between those genes. Some other types of dominance that are encountered, so where you get away from this idea of just, complete dominance like you would see with that first case of a simple Punnett square that we looked at, would be something called incomplete dominance, right? So in this case here, the homozygous dominant, right, in this case has red flowers, right? Homozygous recessive has white. If we were looking at complete dominance, right, you'd expect all of these offspring to have red flowers because all of these offspring would have to carry at least one copy of the gene, right, from the homozygous dominant parent. But instead what you're seeing is something in between, kind of incomplete is pink. But importantly, if you were to cross these offspring, right, then you would see coming again, right, to the surface, a distinctness in the behavior of the dominance as you would see homozygous dominant, homozygous recessive offspring, right? Again, you would see the red and the white flowers, right? But when you have the heterozygous individuals, right, the dominance is somewhere in between. So maybe this is an interaction of the different types of alleles. Maybe this is a dose dependence type of consequence, but it's something in between. But they're still, of course, carrying the dominant and the recessive alleles. And then codominance is a variation on this where you can have multiple different alleles of a particular gene. One example would be the ABO blood typing. That's a favorite for bringing up codominance. So you can have individuals that are type A, right? So they're carrying A alleles for a particular type of gene. You can have people that are type B, so they're carrying the B alleles for a particular type of gene. Or you can have individuals that are carrying recessive versions of both of these types of genes. So basically, the A allele is producing an A type enzyme that's making an A type carbohydrate on the surface of red blood cells. A carrier of the B allele is producing a B type enzyme that's making a B type carbohydrate that's present on the surface of red blood cells. A person that's carrying recessive alleles that's not producing any functional enzyme, is not making any of that type of carbohydrate structure, right, ends up becoming a type O individual. But you can also have individuals that are carrying the A allele, making the A enzyme that produces the A carbohydrate, and they also carry the B allele that produces the B enzyme that makes the B carbohydrate, and their red blood cells express both the A type of carbohydrate and the B type of carbohydrate. They're type AB, so they have a codominance, where both of these dominant alleles are expressing. In terms of evolutionary mechanisms that you may encounter, so what are the different types of things that are driving evolution? Keep in mind that the fuel for evolution is variability. So where do we get genetic variability? We can get genetic variability in a population, shown here on the left, with mutations. Right? So mutations can occur, and if these mutations occur in the germline, right, in the cells that are going to be responsible for producing gametes, then these can be transmitted to the next generation. So mutation is naturally a place where you can get the introduction of more variation for evolution to work on. Migration can be the movement of individuals, right, that contain different variations on a gene from one population to another. Natural selection, right? So if there's variation that exists within a population, right, that variation, right, can lead some individuals to be more fit than others, right? And with that variation in fitness, right, there's going to be a change in the distribution of those alleles within the population. And then genetic drift is where you get an oftentimes kind of chance occurrence, which will come in and adjust the proportion of alleles within a population, right? But it's not due to natural selection or predation or something along those lines. It's some sort of a chance circumstance. But it can still affect the distribution or the abundance of alleles within a population, right? And therefore it can be a mechanism of evolution. As we look at evolution, right, across the kind of spectrum of life, we can map that out with a taxonomic kind of a tree, a phylogenetic tree. And this is a way that it doesn't have to be as broad as this, right? This is bacteria, archaea, and eukaryota. It doesn't have to be this broad. It could be just a handful of species within a genus. But you can map out where these individuals lie. And with this kind of phylogenetic tree, there's information embedded, because it also maps out where individuals within a particular tree, where they share the most recent common ancestor, or where that divergence occurred within the evolutionary tree. And oftentimes, the way that the trees are designed graphically is the distance or the length of these different kind of limbs or branches within the tree is reflective of the amount of time since the divergence. So it can give some information on how long the evolutionary history has been since the divergence occurred, but then also the relationship between members of those individuals that are being represented in the tree, how they kind of are more or less closely related to other members. All right, we'll be going through some sample questions now. All right, with the first one, human skin color, height, and weight depend on the interaction of the products of multiple genes. These and other polygenic traits typically exhibit. And again, with the video, I kind of expect that you'll pause and look at the, if you want, and look at the responses. And then I'll give you a moment to do that, and then I'll jump into the right response. If not, I'll just be kind of moving quickly through the questions. So at this question here, right, it's continuous variation. So most phenotypic outcomes from genetics are complicated. Many genes involved in certain processes with a phenotypic outcome. And because of that complexity, right, there's going to be variation. So there's a lot of genes involved in things like skin color and height, of course, right? So this is going to be continuous variation. Right? Codominance is like the example of ABO blood typing, right, where individuals can be type A with the A allele, type B with the B allele, or they can carry A and B, right? And in that case, both alleles are making enzymes, and both the enzymes are active, but they're making their own unique kind of branching carbohydrate chains that end up on the surface of red blood cells, right? So in this case, they have both the A and the B enzyme making both the A and the B carbohydrates, so they're AB instead of one or the other, right? Or for those individuals that inherit recessive versions of these alleles, they have no enzyme making either of these carbohydrates, right? So they're type O. But because individuals can be A and B, right, they can exhibit codominance. Polyploidy has to do with the number of chromosomes, right? And, a typical case would be like trisomy 21, where an individual has 3 copies of chromosome 21 rather than 2. And then complete dominance is like kind of the typical, very simple Mendelian inheritance of alleles from a gene that shows a dominant version and a recessive version. Pleiotropy, is typical. It's rare that you have a single gene and a single outcome, right? We have tens of trillions of cells that have access to genes, and oftentimes genes and mutations in genes are going to impact various different types of cells and tissues that they're expressed in. So they can have different outcomes in different types of cells and tissues. Which of the following enzymes most likely is responsible for the cleavage of the plasmid shown? There's some illustrations positioned not where they're indicated in the question, just because of the way I edited them for this last version. But this is a restriction endonuclease, right? Ligases cut, helicases unwind, polymerases polymerize, contribute to long growing trains of nucleotides, right? Restriction endonucleases are enzymes that are very specific for regions of and kind of these consensus sequences of DNA that they target, right? So the artist here has even gone through all the trouble of writing down the TTAA and the complementary AATT, right, which is where this enzyme would situate itself and actually go in and hydrolyze the bonds in the sugar phosphate backbone and cut the DNA. The molecules of electron transport chains are found in which of the labeled locations of the organelle represented? This is, of course, a mitochondrion, right? And the components of the electron transport chain are in the inner membrane, C, because they need to be able to pump hydrogen ions, right, as they move electrons through their structures. And those hydrogen ions end up in the inner membrane space. Right, so that's where they get concentrated before being released back into the matrix through the ATP synthase, generating ATP. Which of the following is most likely to increase transcription of a particular gene? The answer here is B. So acetylation of histone proteins. Histones are those protein cores that DNA wraps around, right? It wraps around each one twice. When you take DNA and stretch it out and look at it with a transmission electron microscope, you can see it looks like beads on a string, right? And those beads are these histone cores, these protein cores. And generally speaking, when they get acetylated, they open up the DNA so it's more accessible, right? And you get an increase in gene transcription. Methylation does the opposite. It has kind of loose DNA, what we call euchromatin, more likely to condense into heterochromatin and make the genes less accessible. So it's definitely not methylation. Binding of a ribosome has to do with translation, right? So we're not looking at transcription here. And more rapid pre-messenger RNA processing is also after transcription. And you got DNA gets transcribed into messenger RNA, which is what we're talking about. And then after that, you get messenger RNA processing. So that doesn't make any sense either. Right? And then with the central dogma, then it's translation to protein. Shown above is the summary equation for aerobic cellular respiration. Which of the following indicates a molecule that is oxidized and the molecule that is reduced as a process proceeds? The answer here is B. So sugar is getting oxidized with cellular respiration, right? That's what's getting broken down. And with redox reactions, if something's getting oxidized, something's getting reduced. So here, right, the partner in this equation is oxygen. That's getting reduced. Interphase is a part of the cell cycle that includes which of the following? Cytokinesis, synapsis of homologous chromosomes, replication of DNA, or condensation of chromosomes. And the answer here is replication of DNA. Right, so that's S phase during interphase. Condensation of chromosomes and cytokinesis, those are parts of mitosis. And then synapsis of homologous chromosomes, that occurs with homologous recombination. That's in meiosis, so that's a different process. Which of the following structures is typically found in eukaryotic cells, but not in prokaryotic cells? Right, so that's mitochondria. So when I'm looking at eukaryotic, prokaryotic, I'm thinking membrane-bound organelles. All cells have plasma membranes, so that's in both. Chromosomes and ribosomes, right? Those are integral to the central dogma, so that's in both. It's the mitochondria that are unique to eukaryotic cells. Serum albumin, a blood protein produced by hepatocytes in the liver, has a molecular mass of approximately 66.5 kilodaltons. By which of the following processes is newly synthesized albumin typically transferred from the cytosol of a hepatocyte to the blood? So the answer here is exocytosis, right? We have a cell, it's making something, it's exporting it, that's exocytosis. Phagocytosis, phage is to eat, so phagocytosis is like some immune cells like macrophages. Neutrophils can actually like engulf and eat debris or foreign pathogens, right? That's phagocytosis. So that's not what we're talking about here. Cell lysis, that doesn't really make any sense. That's with a cell breaking apart, right? But that's the way that's phrased. It would be something that you might do in like a laboratory as part of a protocol. It's not even really something that's biologically relevant. Passive diffusion. A protein is way too large and way too charged to just diffuse across a membrane. There's no way it's diffusing across a membrane. Passive diffusion you might get with things like gases, oxygen, carbon dioxide. But yeah, proteins are not diffusing across a membrane. So this is going to take a much more sophisticated process like exocytosis. Which of the following occurs during the processing of eukaryotic pre-messenger RNA molecule? So the answer is D. So the addition of a 5' cap is something that happens during messenger RNA processing, but that has nothing to do with causing transcription to terminate. Splicing is also something that happens with pre-messenger RNA processing, but B doesn't make any sense because exons aren't spliced out. Right? You have exons and introns. I think introns are in between the exons. The exons are what you want. That's the coding region. So the exons are what you keep and the introns are what get jettisoned. This answer suggests that introns are spliced together and somehow encoded. That's not true. And splicing doesn't induce the binding of ribosomal subunits to the initiation codon. Pre-messenger RNA processing occurs in the nucleus, right? And the binding to a ribosome, that's going to happen in the ribosome. In the cytosol. So the answer here is D. All right, the yucca plant cannot be pollinated and produce seeds without the presence of a small white moth called the yucca moth. The yucca moth, in turn, is entirely dependent on the yucca plant for its own reproduction. The interdependence of the plant and the moth exemplifies. The answer here is coevolution. Right? So both this plant and this moth are on their own evolutionary pathways, but these pathways are integrated and intermingled and codependent. So this is coevolution. So convergent evolution is where you would see phenotypes or consequences of evolution in distant kind of taxa. Divergent evolution would be a deviation in different types of traits. And all right, the yucca plant cannot be pollinated and produce seeds without the presence of a small white moth called the yucca moth. The yucca moth, in turn, is entirely dependent on the yucca plant for its own reproduction. The interdependence of the plant and the moth exemplifies. All right, so the answer here is B, coevolution. Right? So both this plant and this moth are on their own evolutionary pathways, but these pathways are integrated and intermingled, and they have a dependency on each other. So these two species are co-evolving. All right, so today we looked at some more topics within some of the different subject areas, and then also went through some sample questions. And as I've mentioned several times already, this isn't a comprehensive review of all things biology, right? It's kind of weaving through some of these different sub-areas to give yourselves an opportunity to think about where you feel really confident and where you need to spend more time preparing. And for preparing, I gave you some resources in the first presentation, so check those out. And with the third presentation, we'll be looking at open response questions, structure and strategy, as well as some more sample questions.